After a eukaryotic cell transcribes a protein coding gene, the RNA transcript called a pre-mRNA is processed. The processing takes place in the nucleus, after which the mature mRNA is released into the cytoplasm. Ribosomes in the cytoplasm translate the mRNA into protein. Here we focus on one of several RNA processing steps called RNA splicing. The pre-mRNAs, just like the genes from which they are transcribed, contain regions that do not code for the translated protein. These non-coding regions, called introns, interrupt the coding regions, called exons, and must be removed. As soon as a pre-mRNA is transcribed, it is quickly bound by several complexes called small nuclear ribonucleoprotein particles, or snRNPs, pronounced SNRPs for short. As the name suggests, SNRPs contain RNAs and proteins. The SNRPs are responsible for splicing introns out of pre-mRNAs. SNRPs bind to sites in a pre-mRNA at or near the intron-exon boundaries. These sites, called consensus sequences, contain nucleotide sequences that are shared by most pre-mRNAs. The SNRPs contain RNA molecules that can bind to these consensus sequences through complementary base pairing. In addition to the SNRPs that have bound to the consensus sequences, other SNRPs, not shown, attach to other sequences within the intron. The SNRPs eventually come together into a large complex called a spliceosome. As the spliceosome forms, the intron loops out. The spliceosome cuts the pre-mRNA at one intron-exon boundary, where it leaves a reactive free hydroxyl group on the exon. The spliceosome uses this hydroxyl group to attack the other end of the intron, and in the process, removes the intron and joins the exons together, forming a mature mRNA molecule. The mRNA leaves the nucleus and is translated into protein within the cytoplasm. The intron that remains is quickly degraded. The SNRPs remain in the cell and are used to splice introns from other RNA molecules.